Okay, so today the idea is to finish off the idea on drought and heat. I'm going to say a little bit about flooding. Friday's lecture, I might have just a few last things to say about flooding, but then most of the session on Friday is a review session for the exam. That will be on Monday. So the exam on Monday starts with the material from C4 forward. Keep in mind that, exam 3 that you've already taken, the level of detail on the light reactions, that's not the level of detail you're getting in this next exam. So you're going to be expected to know the C4 and CAM, but really they're kind of the same pathway. There's just some key comparisons to make between them. So, and then moving into, we got into stress or defense responses. So we'll go through that on Friday as well and think about some of the key ideas that you should be zooming in on, okay? So Friday's session, come prepared to work through some of that material, ask questions, okay? Sound like a plan? Okay. Remember, the exam on Monday will not have any essay. So it will be multiple choice, true, false, might be some drag and drop, I don't know yet, but I'll know more hopefully by Friday, have a better feel for the exam. Okay, so I wanted to say a few last words about drought, which when you look at all of the environmental impacts on plants, drought and heat is a big one. Causes a lot of problems, not just with crop production, but just plants living their lives outside, especially now with global climate shifts going on. That will probably become even more of a problem. I think we left off here looking at the idea that plants do have some ability to try to deal with the situation. And one of them is this making more solutes, playing with their Cy/Sub-S component inside their cells. When you think about the water potential equation, just trying to create a situation where they continue to have water flow into the plant rather than water going out of the plant and into the soil. And certainly stopping transpiration by closing down their stomata, lots of options, but sooner or later it can get to the point where it's the tipping point. It's like no matter what they do, it's not going to work. So if you start to get really severe drought in the soil where there's just really low water flow, yeah, the frictional forces start to really play an impact on that. You'll get to a point eventually where the plants go into a state called permanent wilting. And that permanent wilting point means that they can't, even if you water that plant, they're like done. They can't deal with it anymore. They're pretty much on their last leg. So experimentally, if you force a plant into the permanent wilting point, even if you water them, they won't be able to regain any turgor pressure, no significant movement into the plant. Even if they have their stomata closed, they just can't seem to deal with it in any way. Some experimental plants that they've worked with, this is more dealing with crops, negative 1.5, that's not taking into account the desert adapted plants. They can deal with that, but this is more like crop production plants. So they certainly see an anatomical shifts. The roots will start to shrink. The root hairs get damaged under severe drought. And then they try to thicken up the waxy coating on their roots. So in the root zone, they have the suberin wax is what gets laid down in the roots, or on the roots, I should say. And that definitely, they'll try to increase that and try to thicken it up. But, if it's permanent wilting point, that's pretty much done. And then cavitation, I mentioned that way back during transpiration ideas, when the water is flowing through the plant. If they end up in a drought condition, what tends to happen is air bubbles come out of solution in the xylem because the increase in tension in the xylem cells creates too much of a tension force on that column of water in the xylem. And of course, it's not pure water, it's got gases dissolved in it. So if you put dissolved gases under enough tension, they'll pop out of solution, and that causes an air bubble in the xylem. And that's the fancy word for that is cavitation. It's analogous to like an air embolism in our veins and how that can be deadly for us. Cavitation may or may not kill the plant. It depends how severe and widespread it is. They can deal with it to some extent. So lots of things happening that aren't good when you start pushing the drought into the severe category. This is just a picture of the xylem again to remind you of the idea of, they're empty, hollow cells that will allow for that water flow. And it's the vessel, the wider diameter ones that tend to have cavitation happening more frequently. So that's a huge reason why plants will make clusters of xylem. Just in case some of them do go through that cavitation, hopefully some of the nearby vessels and tracheids will still be functioning. So that's why they cluster their vascular tissue like that. Hedge their bets against cavitation issues. So up above ground, there's also shifts going on during severe drought. You tend to see the leaves will actually start to shift their morphology in response to the extreme drought and heat. Under heat effects in particular, you tend to see, and this even goes into thinking about desert adapted plants, how you think about some of those plants that can deal with consistent heat, and it's not just a freak event. You tend to see, in general, if a plant's been exposed to prolonged heat, they'll end up making smaller leaves. Their leaf size can reduce. So even a plant that is not desert adapted, they can shift their leaf morphology. If they are getting consistently hot, dry conditions, they will end up making leaves that are smaller. And they'll also kind of make the morphology of the leaf surface. They'll try to make sure that their boundary layer is nice and thick. So the boundary layer is just that layer of still air that's around the leaf. We have a boundary layer around our bodies as well. It's just that layer of relatively unstirred, relatively still air molecules. So if the plant can like tuck their stomata into those stomatal crypts, or somehow create a situation where they have a nice thick boundary layer right around where their stomata are, that will slow down their transpiration rate. And the small size of the leaf, it helps drive off the heat off the leaf faster. Shape. So not only the size, but the shape of the leaf. They tend to morph into a design that can promote smaller surface area so that they're just, again, not exposing as much of their body to the heat and trying to minimize heat loss or water loss. Another strategy is that the leaves that will get produced under prolonged heat, even if the plant normally doesn't have a lot of trichomes, they can switch into trichome production. They have seen that experimentally where you can have the leaves ramp up their trichomes. And the reason they're doing that is that it bounces the sunlight energy around better so that they're not getting heated up as much. And it just minimizes transpiration rate. It slows it down. It creates more of a physical barrier for air, wind flow across the leaf. So if you can slow down any of that stirred up air around the leaf, it will slow down transpiration. So some interesting changes that they can make with their leaves. So even on the same plant, when it's putting out new leaves, they can make these changes. Here's just a visual trying to get across that point where you have the shift in the leaf morphology. The wild type is on the left and prolonged heat. Or no, I have that backwards. The one on the left is the one better adapted for cooling. Okay. Above ground too, they can try to ramp up their wax levels. And again, that's just try to prevent water loss, slow down transpiration, it bounces light off them better. Hopefully their leaves won't heat up as much. And again, if you can, excuse me, if you can hide away your stomata and tuck them away so that the wind isn't directly coursing right across the stomata, that will slow down transpiration as well. So go ahead and sink the stomata or hide them in those stomatal crypts. And that's basically just creating a nice thick boundary layer, that layer of still air that will slow down the transpiration. Even the idea of how the leaves are oriented in three-dimensional space under high heat and drought makes a difference. And so often you'll see if the leaves are getting too much sunlight energy, they will shift their leaf blade so that it's not 90 degrees to the incoming sun. And so that's normally what they do. They try to be 90 degrees. So if you're thinking about high noon where the sun's directly overhead, they'll try to have their leaves horizontal to the ground essentially. So it's a 90-degree exposure to the incoming sunlight. Well, if you're a plant that's experiencing a lot of drought in high heat, you'll actually try to shift your leaf blade so it's more upright. If you think about high noon, you'd want your leaf more upright, vertical, so that as the sunlight energy is flowing down onto the plant body, it's actually a lot of it's going past the leaves and not striking the leaves. And so the angle so they can manipulate their petiole, essentially the leaf blade stem, they can take that petiole and cause movements to occur. And so generally they try to kind of hide away from the sun if they can to minimize how much sunlight energy is striking the leaf and heating it up. You may have noticed if you've been out hiking in grasses, fields of grasses, that you'll see on days that are, if there's been a period of drought, you'll actually see the leaves roll up lengthwise. So, the long leaf blade, they'll just kind of roll up on themselves. And so, again, that's just trying to minimize how much of their leaf blade is exposed to that high heat and intense sunlight. And so I've noticed that many times when I've been out hiking in the open field, that's when we've had drought, it'll do that really interesting roll pattern. And then just wilting, that's a response certainly to lack of water, but it does change the angle of the leaf blade to the sun. And so it can actually be an adaptive response that the plant's doing in order to try to minimize direct sunlight energy onto the leaf blade. So you'll see that with different plants, depending on the plant, the leaf design to begin with, they'll either have their leaves go more upright or they may droop them down. And it's kind of depends on the species. So here you're looking at a well-watered soybean, but if you put them under stress, you can see how this one's actually doing both. It has one leaf blade up, one leaf blade down, so that they're trying to do more parallel position rather than horizontal to the light. And then again, when under severe stress, but that position of the leaves, even though you'd say, oh, it's wilting, yes, but it's also beneficial response in the sense that it does allow for the sunlight energy to just flow by the leaf blade. So above ground and below ground, plants are busy trying to minimize issues with water loss, too much heat. Here's a schematic just showing the idea that when the sunlight energy is hitting the leaf, certainly, some of that is going for photosynthesis, but if you are under high heat conditions, you do have some of that sunlight energy getting dissipated. Some of it will bounce off in longer wavelengths and that will come off as heat. You also have conduction and convection currents around the leaf blade that will stir up air and they do say that it can cool the leaf to some extent. And then, of course, the plant is busy doing transpiration. So the evaporation idea is when the plant is doing transpiration. So they have several ways to try to kind of spill off this extra heat and try to make sure that their leaves aren't getting too heated up. So desert adapted plants kind of do this routinely. Desert adapted plants, most of them, if you think about it, they don't really have giant leaf blades. They're small leaves. They're often very thick leaves and more roundish shape to the leaves, very thick cuticle. And so if you kind of think about what do desert adapted plants do, those plants that aren't desert adapted, they can still try to adapt some of those mechanisms when they are under the drought and heat. And then if you remove that pressure, if you start to give them lots of water and the heat comes down, you'll see them revert back to how they normally grow. So they're very capable of flipping back and forth if need be. Plants are awesome that way. They have a great ability to adapt very quickly. Okay. So that's it on the drought and heat. And I wanted to kind of flip the coin and talk a little bit about, okay, what happens if you do the opposite and flood? And there's a couple of key points I wanted to mention about that. Flooding is an issue, and it's not going away. Flooding is still a thing. They are predicting global climate changes are going to make it worse in some areas of the world. So let me share that slide that's posted. So the big thing to think about with flooding is the roots, those poor roots. Roots need to have oxygen, just like we need oxygen, roots need oxygen. They have their own version of doing aerobic respiration. And if you can't get oxygen to the roots will start to die. And obviously, if the roots start to die, the whole plant is at risk. So I just put this visual here to get you thinking about the mitochondria. We're zooming in there, thinking about how the mitochondria is allowing for ATP production through the use of oxygen. And so aerobic respiration is all cooking along inside the mitochondria. I'm not going to ask you to know any details about this. I just put this up for those of you that are visual learners. Again, we're talking about an electron transport chain process. And this is all happening in the mitochondria. And here in the lower right, you can see oxygen coming into play. So oxygen is the terminal electron acceptor, and it's there's this electron transport chain process that is linked to proton pumping, and then that allows for ATP synthesis. So it is a process that's cooking along in your mitochondria right now. Plants do it a little differently. They have some extra players in the game here, but it's the same basic premise of an electron transport chain process linked to proton pumping, which leads to a proton gradient, which leads to ATP synthesis. So all aerobic organisms need to do this to stay alive, and the roots are no exception. So if you think about soil that is a more typical soil that's well drained versus a flooded soil, really the huge difference that impacts plants is the access to oxygen, essentially. And so aerobic means that you have oxygen there. Another way to say that would be oxic. That means there's oxygen there. And then the other flip side is anaerobic or anoxic, meaning no oxygen. So anytime you see AN on the front of the word, that's without. So without air, without an oxic aerobic environment. The other thing to keep in mind, even if you have a well-drained soil, there's still issues. You have to realize that oxygen dissolved in water diffuses pretty slowly. It's not a rapid thing by any stretch. So there is that limitation that's always on the plant in terms of its ability to get access to oxygen. But it's very severe in a flooded soil because now the oxygen is all in the water solution, and it really slows down access. So there's definitely plants that exhibit capabilities to adapt and others that really are not so good at it. So flood sensitive plants is how they refer to the ones where experimentally they say, let's flood the plant for 24 hours. And if they, if that results in a crash in their biomass, then we'll call them flooding sensitive. And then flooding tolerant are, you can put them under a flood condition and they'll bounce back from it. Adapted plants are the ones that deal with this on a regular basis. So you think about plants that are along the areas of high tide, low tide in the ocean areas where you have, and then of course swamp plants that have those issues too. So there's different ranges of how plants are going to be impacted by this. So I just wanted to mention a few things about how do plants adapt to this, and then what could be some problems in addition to just lack of oxygen and suffocating. What else could be going on that would impact the plant? So the plants can adapt physically by shifting their root morphology. They can actually, and their stems too, if the stems are flooded. You can have they'll go through planned cell death. They'll go through apoptosis in different regions of their body that are under the flood. And it ends up creating a new tissue morphology that allows for faster diffusion of oxygen through the tissue. So it's just trying to get more ways for oxygen to openly diffuse through air gaps in the tissue rather than having it move through from cell to cell via the cytoplasm, for example. So there's a fancy name for it. They call it parenchyma tissue. So parenchyma, A-E-R, is referring to oxygen. I'm just saying it's tissue that is designed to allow for better oxygen flow through that part of the plant body. And so when you look at it under the microscope, it just looks like big holes in the tissue. It's just open air spaces in the tissue. And they know that ethylene is actually responsible for creating that. And so when the plant is under a flood, you'll see an upswing in ethylene, and it'll actually tell those regions of the plant, it'll signal to those regions, go ahead and die off parts of your cells so that we have bigger gaps, bigger air gaps. And so they actually give the kind of the suicide notification, go ahead and kill off sections of your body so that you can get those air gaps in the tissue. And then if you take these plants and experimentally then stop the flooding, they can revert back. And so new growth can revert back to their normal root growth where you don't have as much parenchyma tissue. So it's really amazing how quickly they can respond. So on the left, you see the wild type without flooding event. And then on the right, you can see far more big gaps of, there's just no tissue there. It's all air gaps. So that's just better for oxygen to flow through. So plants are pretty good at doing that under flooding conditions, and they can start that process pretty quickly because ethylene is a gas, so that can flow through the root tissue and reach distant parts of the root tissue even. So that's a good thing. The other huge impact that you see with what's going on in a flooding soil is the microbes in the soil that are associated with the plant. You can get a shift in who's there in a bad way. And so there are plenty of microbes in the soil that are just fine without oxygen. They don't care. And in fact, some of them are poisoned by the presence of oxygen. So this diagram I actually showed in my other class, where this is the idea on the left of aerobic respiration, where you have oxygen being used to accept electrons. On the right is how some microbes do it in the soil, where instead of using oxygen, if they can't find oxygen, they'll say, oh, all right, I'll use nitrate instead. So NO3 minus is being used as an electron acceptor. So microbes are busy doing that. Well, plants love nitrate. So if microbes are busy doing this in anaerobic conditions, they're scarfing up the nitrate. The plants aren't getting it. So you get now a reduction in nitrogen availability to the plant. So that's a big deal. And there's a lot of microbes that can do this right around the root zone. And so it's essentially stealing that lovely nitrate that plants need, and the microbes are using it instead, all because they're denied oxygen due to flooding. If oxygen was there, they'd pick oxygen. It's better to use oxygen. But if oxygen is not available, then they'll switch to nitrate. Well, now the plant's getting starved of nitrate. So what I showed you there on the previous slide is showing nitrate accepting an electron here and getting reduced to nitrite. And some microbes do that. And then some microbes can keep going. And the big picture idea here is they can take nitrate and push it all the way to gas form, possibly all the way to dinitrogen. So these last three molecules are all gaseous, and so those tend to go up into the atmosphere, out of the soil, into the atmosphere. So again, this is just pushing nitrogen out of the soil that the plant can't access now. Because once it gets put into dinitrogen, plants can't touch it. You have to have a microbe grab the dinitrogen and fix it. And so again, if flooding is going on, this can ramp up in high levels to the point where now that soil is getting depleted of nitrogen that the plant could have used, but the microbes are using it instead. So that's a big problem. And so you get that loss of nitrogen from the soil, so the plant can't get that. Another thing that you have happening in the soil under anaerobic conditions is the iron shifts to, if you shift the iron form from ferric, 3+ is ferric, Fe2+ is ferrous. You have both of those forms in the soil naturally. Ferric form is a little more challenging to get access to. Ferrous is more soluble. So what they actually see is it becomes too soluble under flooding, and it gets to the point where the plant gets toxic exposure to ferrous, and it actually creates toxicity to the plant. So it's a little bit too much of the iron now. Another thing that's going on is that under anaerobic conditions, you'll have in the soil microbes busy. They'll take sulfate, SO4(2-), and they'll reduce it to hydrogen sulfide, which is a gas, and that can percolate out of the soil. But it's important to realize that all living organisms are poisoned by hydrogen sulfide, including plants, including us. If we got exposed to that to high levels, we'd die. It's a poison. And so, again, having that around your roots when you're already getting starved of oxygen is a really bad situation. Another thing that's happening via the microbes in the soil is under anaerobic conditions, if they don't have oxygen, some of the other things that microbes can do is fermentation. They'll run a bunch of fermentation reactions. Well, some of them, when they do that, one of the fermentation products is butyric acid and acetic acid. If you get those at high enough levels, those are also toxic to plants. So basically, you see a situation where, yeah, flooding is bad because it reduces oxygen, for sure. But now you have the microbes in the soil, you have the wrong kinds of microbes starting to gain their population sizes go up, because now they're like, oh, anaerobic environments, yeah, this is my game, I can do this. And so their populations start to go up, all the aerobic microbes start to die off. And so you get this shift in who's there around the roots to the point of they're not very good for the plant. They're doing all of this that's being shown here on the PowerPoint, and the overall impact on the plant is very negative. So it's not just the lack of oxygen, it's all this other stuff that gets piled on that's impacting the plant. By the way, the plants can do fermentation. If they get denied oxygen, they can switch to some fermentation pathways, but it's really inefficient, and they can't do that forever. And so they do have mechanisms where they can hang in for a while, but it's just not the best idea. So I'm not going to go into this. This is just for those of you who are visual. This is the idea of fermentation. And so the green box in particular that you see here, and then I blew it up here on the top right, that's the end of the fermentation pathway. So they can do this, but again, it doesn't get you much ATP, and so their tissues will start to die off if that's the only thing they can do. It's a very modest ability. It's only 2 ATP that they can create through that fermentation pathway. So that's not the best, and sooner or later that's not going to be enough. And then the roots under flooded conditions, it just creates a condition where it's really hard for them to have mineral uptake. And even though there's plenty of water there, they're having a really hard time taking up the necessary minerals. I'm not going to go into any details on that. It's just the big picture idea on that. And then another thing that they see, which is kind of weird when you see it with your own eyes, what you see happening under flooded conditions is it will trigger ACC synthesis. Well, that's my pointer. So ACC is the yellow one here in the yellow box. And why that's important is it can lead to ethylene. Ethylene, of course, is one of the gaseous hormones. So what they see happening under flooded conditions is that you get in the shoots this upswing in ACC, which creates ethylene production in the shoots. And so it stimulates the shoots to go through that epinasty growth pattern that you saw in lab, where you have the leaves and the petioles bending down. They look like they're wilting. And so it's really weird because you'll look at a flooded plant and you'll say, oh, it's wilting, but it's really not wilting. It's the epinasty. It has plenty of water. It has way too much water, actually. So it's not wilting from lack of water. It's just the opposite. And it's the epinasty experience that you're seeing. And so they aren't quite sure why plants do that, but it's a kind of a common phenomenon under flooding. Their shoots will go through epinasty, and that's because of the production of ACC, and then from that you'll get ethylene. And so flooding versus non-flooding, they're just trying to show that you get this big upswing in ethylene. And then when that's in the shoots, if that's all going on in the shoots, you'll see that epinasty experience. So kind of a weird dynamic. One other thing that plants can do, although sometimes it can boomerang on them and cause problems, is if you can imagine being a plant and you're slowly getting flooded and the water level keeps rising and the water level keeps rising, one way that you could try to deal with it is try to get out of it, right? Now they don't have legs, they can't walk away. What could they do to try to get out of the water? They just try to grow taller. And they actually see that. A lot of plants will do this. They'll start to elongate their stems. So they'll start to put a lot of energy into, let's get taller. So hopefully we can reach above the water. We can get enough of our body above the water that we aren't going to suffocate and die. And so they actually will do that. And it can be a pretty rapid response. They'll start to increase their stem tissue, get bigger and bigger and bigger. And they refer to it as kind of an escape strategy. They don't have legs, they can't walk away, but at least they're trying to physically move away just through this growth pattern. And so we learned about that, when you think about the hormones involved, we're thinking about gibberellic acid, right? That's the one that can really make the stem elongation occur. You did that lab with the dwarf pea versus the wild type pea. And so you can definitely see how that really impacts the stem tissue. It really can create some really decent stem tissue growth. So here, experimentally, they were looking at rice plants and looking at the idea of wild type versus genetically modified. And so here, if you look at the figures on the right, what they're showing, if you look at the first one and then the fourth one, those are like the wild type ones, or the ones that weren't genetically modified to try to handle flooding. So you can see as you start at the top, that's like before they got flooded. And then you can see the middle and bottom picture, that's 16 days and 7 days. Oh, that's recovery. So 16 days after submergence is the middle picture. And you can see that the first and fourth plant has kind of died. They're not doing well at all, but the other ones that were genetically modified are doing okay. The fourth one is maybe a little tricky there. And then you see on the very bottom that 7 days, so a week after they stopped flooding and allowed them to recover, you can see how the ones that were genetically modified made it. And so what they hypothesized was happening is the wild type ones, the intolerant plants, they did that escape strategy. They tried to escape, but they didn't do it carefully enough in the sense that their stems were too weak. So when the water moved away, their tissues couldn't handle that loss of support of the water. They ended up flopping over and dying. Whereas the other ones did the escape strategy, but not so dramatically. And after the water was removed, they were able to deal with it and survive. So what they were playing around with was the gibberellic acid. They were saying that the modified ones, they made it so that they weren't overdoing their stem growth. So they were a little less sensitive to the gibberellic acid. So they grew, but they didn't do it extreme like the first and fourth plant did. So they're playing around with that, looking at ways to try to help plants prolong their lives through flooding and hopefully make it all the way through. And then once the flood is gone, you don't want the plants flopping over and dying. Obviously, you want them to keep going. So that's some interesting research that's going on. And so the flooding is kind of complicated because it's not just the lack of oxygen. That's certainly a huge deal, but it's also all the other stuff that kicks into action in the soil, and it's primarily the microbes that are doing some damage there with the products they're creating and the toxins they're creating, and then using up all the nitrogen. Okay, so that's really all I want to say about flooding. I guess we're done with that. So basically we're done with the slide sets for the semester. So I could actually start right now kind of getting into the exam material, just kind of starting on, if we go back into some of the previous slide sets. Okay. Ugh, we gotta get rid of that. Okay, let me. I think so. Yeah. Let's see if I can get this to work. Okay, so this is the carbon fixation, but we're done with most of it. So the C4. Okay, so the slide, it says, why do the PCO cycle? So the content after is where the exam content starts. And so we looked at the C4 cycle as an adaptation to deal with the PCO cycle. They're trying to minimize it. And so we looked at the C4 cycle and then the CAM cycle, and both of those are being used by plants that are adapted to areas that are consistently hot. In C4 plants, you're thinking tropical areas, subtropical, consistently hot. Yes, they get some rain events, but they have some drought too. And then CAM plants, we're pushing it further into desert adapted, where they're just consistently hot, very little rain. And so we were comparing those two cycles, and there's a lot of similarities actually when you think about the two cycles. There's so this is the C4 looking at the idea where you don't even have rubisco coming into play at first. That enzyme isn't involved at the start. And so again, if you're trying to keep oxygen from rubisco, this is one mechanism to do it. They hide away rubisco. They put rubisco in a portion of the plant leaf tissue, so it's all being done in the bundle sheath pretty much. Okay, so they kind of sequester away where rubisco is. And in that area, they don't really do a lot of the light reactions there. So that means not much oxygen is getting made in that area. So rubisco is kind of physically sheltered because it's in the bundle sheath. And then you have the rest of the leaf cells, the mesophyll cells of the leaf. You see here the carbon dioxide coming in, and it works with the pepcase enzyme. That's the one enzyme that kicks it off, which is good because pepcase could care less about oxygen. And so there's no fear of running the PCO cycle in this condition. And so the CO2 comes in into the active site of the PEP case and then reacts, PEP with the CO2. And then you end up eventually with malate. First it's oxaloacetate, then you get malate. And now it's like, okay, now we have to get that CO2. It's in the malate molecule. That's where it is now. It's stuck onto that. So now we got to get that CO2 delivered to Rubisco. So that means you got to shift it over into the bundle sheet. So when you think about, what's going on there, it's just a mechanism in play. So here again, you see the CO2 coming in, pepcase enzyme reacting CO2 and PEP, and then you get oxaloacetate to malate. And so that CO2 is part of the malate molecule, and malate's kind of like the vector now. It needs to deliver that CO2 over to Rubisco. And so here you see that happening where the CO2 is getting cleaved off the malate. So it's just a mechanism to make sure Rubisco is not seeing a lot of oxygen, because if Rubisco sees oxygen, it's going to run the PCO. And the PCO cycle is not generally a good thing to keep going. So when you look at the C4, it's a physical way that they're dealing with it. They're saying, let me kick off the C4 in the mesophyll, use an enzyme that care less about oxygen, and then get the CO2 into the malate and have malate do the delivery into the bundle. So it's this physical game that they're playing to minimize Rubisco seeing too much oxygen. So you can see if it keeps happening over and over and over, they'll keep getting CO2 kind of flooding Rubisco, making sure that it just sees a bunch of CO2. Are there questions on that idea? Is that sounding vaguely familiar? Yes. Yes, a fraction of it does occur back in the mesophyll. So most of it occurs in the bundle sheet. But the part that of the Calvin cycle that requires ATP and NADPH, the reduction phase, they actually stick that back into the mesophyll. Because in order to get ATP and NADPH, you need to run the light reactions. And the bundle sheet is like, no light reactions. I don't want them here. That will generate oxygen. And so a part of the Calvin cycle does shift back here, just that reduction part, and then everything else. So the carboxylation part and the regeneration part all take place. So the bundle sheet is trying to say, let's not do light reactions in the bundle sheet, because then that produces oxygen. And Rubisco, we don't want Rubisco seeing oxygen. So. Other questions on the C4 cycle?
I'm looking forward to know, obviously, the intermediates and how this transfers to the next cell.
Yes, and so know the terminology too, please. So the carboxylation, decarboxylation, regeneration, because I'll be phrasing it that way. So that, let's go back to the image. I can get my computer to behave. So carboxylation is just this CO2 coming in and reacting with PEP. That's carboxylation. And then decarboxylation is when malate makes it over into the bundle sheet. And decarboxylate means you're getting a CO2 coming off of malate, getting cleaved off and fed to Rubisco. And then regeneration is talking about the idea that you need to get more PEP made so that you can keep this whole thing going. So please learn it so you know those three terms, because that's how I'll refer to it when I ask questions. And then the CAM is similar. Most of the same players are in play. So it's not a brand new cycle you have to learn. It's just that they've done it on a day-night rhythm instead of a physical separation of where things are happening. It all happens in the same cell. It's just they do a day-night rhythm. So they'll all night long, when it's much cooler outside and the humidity is raised up a little bit, they can risk opening their stomata to take in the CO2, but because you can't run the Calvin cycle at night, it just won't happen. Those enzymes need light. So the Calvin cycle is not running, so they have to do something with that CO2. So they'll stuff it in their vacuole and save it for the day. And then when the daylight, the sunrise comes, strikes the plant body, then they're like, okay, now we can do photosynthesis. And so then they'll move the malate out of the vacuole and get it to the Calvin cycle and cleave off that CO2. And so it's a really neat adaptation to just minimize, open your stomata at night. Don't do it during the day. That's kind of a really risky procedure. So do it at night. But because they can't run the Calvin cycle, they have to store it. And so we'll just, put it in the vacuole. And then during the day, they can go ahead and get the Calvin cycle running. So a lot of the same players. Really, the key difference is that you have starch is the source of when you look at the phosphoenolpyruvate, it's coming actually from the breakdown of starch. And so you have that kind of difference, but it's the same enzyme. Pepcase is working with the CO2 as it comes in. It's reacting it with PEP. You get oxaloacetate again, which turns to malate. So same players in that sense. Questions on the CAM pathway? Right. And so that procedure is so good at making sure Rubisco just is getting flooded with CO2, lots of CO2. So Rubisco doesn't need to worry about, oxygen really isn't coming in high levels there. And then the only thing we really said about sucrose, we didn't really say a whole much about that. It's important that you understand that you have, if the plant says, you know what, I need some sucrose, it's not happening in the chloroplast. They will mobilize that out into the cytoplasm so that G3P, once the Calvin cycle's cooking along and you get that pool of G3P molecules, some of those will get exported for sucrose. And when we say export, we mean it's going into the cytoplasm. But that's really, I didn't ask you to know in detail these pathways. It's just understand that if the plant says, I want to make sucrose, has to export it out, and it's tightly controlled. You have this transmembrane protein that's going to control that process. There's a lot of other controls on it too, but the only one I really talked about was the protein in the membrane of the chloroplast that will allow for the G3P to come out, usually in the form of DHAP, but basically you have some of that G3P getting sent out through that translocator, and it's this one-to-one process where the G3P comes out as the phosphate comes in. And so it's a one-to-one exchange process, and that's one of the ways they control it. So they can control whether or not they're going to get sucrose. So it's an either or. Either the plant will use that G3P to make starch, keep it in the chloroplast and make starch, or it might send out some of it to go ahead and make sucrose. But it's tightly controlled at many levels, including that transmembrane protein. So you got to get G3P coming out, inorganic phosphate coming in. So that's going back. This is the Calvin cycle. This is, where you see that export idea on the far right. That's what they're talking about there. So that's that slide set. So that is on my slide set. It looks like it's slide 40 is where the C4 cycle starts, I think. So it's right after the slide that says, why do the PCO cycle? So in general, the only thing I'm going to ask you about the PCO cycle is kind of indirectly. Like I'll say, why would a plant do the C4 pathway? Like you need to understand it's an adaptation. They're trying to minimize the PCO cycle. But that's really the only way PCO is going to show up. It would be if I ask you why. Why is the C4? Why is the CAM happening? But otherwise we're done with the PCO. Any questions on that slide set? Okay, well, on Friday, after you review this slide set, if you have more questions on it, we can certainly pull this up again. But on Friday, I'll also go over the other slide sets and get you thinking about key points